Welcome everyone to today's investor update, which has been timed to coincide with release of Amplia's March quarter Appendix 4C and quarterly update. Before we proceed, and in the spirit of reconciliation, Amplia acknowledges the traditional custodians of country throughout Australia and their connections to land, sea and community. We pay our respect to their elders past, present and emerging, and extend that same respect to all Aboriginal and Torres Strait Islander peoples listening today. The first few months of 2021 have been pretty exciting ones for Amplia, so we thought it would be useful to summarise some of the key events in a presentation, as well as provide the opportunity for shareholders to ask questions. During today's presentation, I'll touch on the 4C first of all, then step through the presentation, which was released to the ASX this morning, Melbourne time. I invite you to submit questions during the presentation using the Q&A function in Zoom, and I'll do my best to respond to these after the presentation. So in reference to our 4C quarterly cash statement, we reported that at the end of March, the company held $1.8 million in cash. And during the reporting period, we consumed $996,000, of which 2/3 was dedicated to research and development, primarily associated with our phase 1 clinical trial of AMP945. In the coming quarter, expenditure is expected to decrease as the phase 1 trial enters its reporting period. Please take note of our disclaimer. Just to align us all, first of all, on Amplia's focus areas, we're developing new drugs which inhibit focal adhesion kinase, or FAK. The reason we're targeting FAK is because it plays a central role in two significant disease areas, cancer and fibrosis. In fibrosis, we expect that our FAK inhibitors will be used as single-agent antifibrotic drugs. And in cancer, we expect that our FAK inhibitors will be used as combination therapies alongside other chemotherapies, in particular in cancers where fibrosis plays a role to support tumour growth, such as in pancreatic and ovarian cancers. The two indications we're most interested in at present are pulmonary fibrosis, which is a devastating progressive disease with relatively few and rather poor treatment options at present. And we're also focused on pancreatic cancer, which is a very dangerous form of cancer. Patients with metastatic disease who are treated with the best therapies currently available have a median survival time of around about 6 to 8 months. And again, the link between these two therapeutic areas is that they're both in some way fibrotic diseases. AMP945 is our lead development candidate, and in order to address these two therapeutic areas with minimal duplication of effort, we've designed our development plan to be flexible and support both therapeutic areas. This has taken us to where we are today, where we're nearing completion of a phase 1 clinical trial of AMP945 in healthy volunteers. We're now moving into the planning stages for distinct phase 2 clinical trials in both pancreatic cancer and pulmonary fibrosis. So I'd like to provide you with a short update on the phase 1 clinical trial. The trial commenced in October 2020 and completed dosing this month. It was conducted in healthy volunteers at a single site here in Melbourne, namely at Nucleus Network. The trial included a number of components that I'll address in more detail on the next slide. But it is worth noting that dosing in the trial was completed both on time and on budget. This slide gives you an idea of how the phase 1 trial has rolled out over the last 6 or so months. We started by looking at single ascending doses of AMP945 and then moved into multiple ascending doses. All the while, we were looking at in the pharmacokinetic studies at how well AMP945 is absorbed after oral dosing. We also looked at whether food affects the absorption of AMP945, and we're currently looking in the pharmacodynamic study for evidence of interaction of AMP945 with its intended target, FAK. So far, we've found that AMP945 appears safe and well tolerated at all the doses we've tested. There has been no evidence of any food effect on the absorption of AMP945, and the pharmacokinetics that we've seen in humans supports once daily oral dosing of AMP945. Our final reviews of the data are ongoing, but based on the safety, tolerability, and pharmacokinetic data that we've seen so far, we're moving ahead with our plans for phase 2 clinical trials. We expect to receive the final study report for this clinical trial later this quarter. Switching gear to talk about the Garvin Collaboration, which we announced last month. So over the last year or so, Amplia has been collaborating with Professor Paul Timpson and his colleagues at the Garvin Institute of Medical Research in Sydney. Paul's a preeminent researcher in FAC biology with a keen focus on having his work translated into clinical practice. And his research group has spent the last 6 or 7 years looking at the effect of FAC inhibition on pancreatic cancer in models of that disease. The key finding of Paul's group has been that when done in the right way, inhibition of FAC both impacted the fibrotic tissues surrounding pancreatic cancer and can augment the activity of standards of care in models of pancreatic cancer. So we became really interested in Paul's work and asked if we could test our clinical candidate, AMP945, in his models. When Paul's lab did this, they were able to confirm that AMP945 exerted the same effects as the reference compound, was clearly antifibrotic, and also augmented the impact of standard of care treatments in the model. So based on these exciting initial results, we agreed to establish a more formal collaboration between Amplia and the Garvin Institute to both build on our initial findings as well as explore new opportunities. I just want to spend a few minutes talking about some of the exciting data that we've received from Paul Timpson's lab. You may have noticed this slide in a previous presentation that we released in February this year. In this experiment, we looked at whether AMP945 could inhibit the production of collagen, which is a key building block of fibrotic tissues. And the data shows that in vitro, increasing concentrations of AMP945 decrease the amount of collagen produced by fibroblasts. The data shown on this slide, which was also reported in February, shows that increasing concentrations of AMP945 inhibit remodeling of collagen into these tight, ropey, fibrotic bundles that form the tough scar or fibrotic tissue. From here on, we've not presented this data before now. So one of the really clever things that Paul Timpson's group can do is go look inside tumours in a living mouse and observe the biology as it's happening while that mouse is alive. And this slide shows a typical experiment where tumours are first implanted under the skin of a mouse. They're allowed to grow for a few days, and then AMP945 in this case was dosed orally for 4 days. After this, we look inside the tumour in the living mouse using special imaging technology and see what the FAC inhibitor did within the tumour. First question we wanted to answer, of course, is did AMP945 inhibit its intended target, FAC, inside the tumour? And this slide tells us that it did. We can measure the levels of activated FAC in this model using fluorescence of the cells within the tumour. And on these two panels, warm colours towards the end of the red spectrum, towards the red end of the spectrum, indicate relatively high levels of FAC. And cool colours towards the blue end of the spectrum indicate low levels of active FAC. The cells on the left panel are in untreated mice, and those on the right are in mice treated with AMP945. And if we quantify the intensity of these colours and the end of the spectrum that they lie closest to, we can detect a significant difference in active FAC between mice that are treated and untreated. So AMP945 is hitting the target it's intended to hit in these living mice. The next question that we wanted to ask was, if we are inhibiting FAC, are we getting the effects that we would like to see as a result of that? Specifically, are we seeing the downstream impact on fibrotic processes? And this slide shows data that tells us that we are most certainly seeing effects on fibrosis. We see a depletion in both total collagen and cross-linked collagen. As we've already mentioned, these are key building blocks in fibrosis. So again, this result that we're now seeing in vivo recapitulates the results that we had seen earlier in vitro. Finally, we did another experiment to see whether AMP945 was able to augment the effects of gemcitabine and Abraxane, which is a standard of care in pancreatic cancer. In this experiment, Paul's lab established tumours in mice and then treated those mice orally with AMP945 for 4 days. They then stopped treatment with AMP945 and on the following day, treated mice with a single shot of standard of care gemcitabine and Abraxane. A day later, they removed the tumours and looked at the two key markers of tumour health within those tumours. One of these markers is called cleaved caspase 3, and it's a marker of tumour cell death. After treatment with AMP945 and gemcitabine and Abraxane, we saw a significantly higher level of cleaved caspase 3 in these tumours, which told us that more tumours in these mice are dying. Another marker is called Chi67, and it's a marker of proliferation of tumour cells, in a sense the opposite of cleaved caspase. After treatment with AMP945 and gemcitabine and Abraxane, we saw a significantly lower level of Chi67 in these tumours, telling us that tumour cells in these mice are quite sick and are not as proliferative. So overall, the results from the work with the Timpson group at the Garvin Institute so far has told us that AMP945 inhibits fibrosis markers both in vivo and in vitro. Oral doses of AMP945 in mice inhibit the intended target, and priming with AMP945 enhances the effect of gemcitabine and Abraxane, as measured by impact on markers of cell death and proliferation. Finally, I'd like to talk about some much more recent data that we've received, where we've been looking at the impact of our FAK inhibitors in a model of NASH, which is a fibrotic liver disease. So NASH is a progressive disease that affects up to 5% of the human population. It's a serious disease for which there are currently no decent therapeutic options. The disease progresses through stages of steatosis or fatty liver to inflammation and then onto fibrosis. And like many fibrotic illnesses of the liver, can progress to hepatocellular carcinoma or liver cancer. There's several models of NASH that are used preclinically, and we selected the so-called STAM mouse model. This model is preferred by industry because it's believed to best recapitulate the human disease. In the model, NASH develops in these mice over a period of about 7 to 8 weeks. We tested both AMP945 in the AMP886 and AMP945 in the STAM model, and we're really excited to see that both molecules were able to impact key markers of disease. In the left panels, we see that as a result of treating with AMP886 and 945, we see suppression of drivers of the disease through depletion of both the number of fibroblasts, the cells that produce collagen and create fibrotic tissue, as well as the level of activity of those fibroblasts. In the right-hand panels, we can see both FAK inhibitors reduce NASH symptoms and levels of fibrosis as reflected in the composite NAS score, which is a composite score measured to develop to measure the level of inflammation in the liver leading to fibrosis, as well as impacting the levels of fibrosis measured by collagen staining. So these data are exciting to us, not only because they show us that our FAK inhibitors may offer potential in NASH itself, but they also provide further partnering potential and reiterate the broad spectrum antifibrotic activity of our FAK inhibitors. So the take-home messages from the NASH study so far are 945 and 886 significantly inhibit fibroblasts and their activation in the liver. These effects translate into inhibition of NASH itself and fibrosis. And the findings support the utility of our FAK inhibitors in a variety of fibrotic diseases with unmet medical need. Just to summarise then, we feel that that Amplia has made excellent progress on development of its FAK assets. The phase 1 trial is on track. We've received exciting data from the Garvin Collaboration and early signs of efficacy in the NASH model, which is a completely new indication and broadens our commercial and partnering potential. We believe that the company's planned phase 2 studies are really well supported now by a platform of both clinical and preclinical data. And we expect further transformation and growth in the company as the company readies for phase 2 clinical studies over the next few months. That's the end of the presentation now, so I'll take a look to see if there are any questions that have been received. I have a question here. What further data do you expect from the Garvin Collaboration? One of the things that the Garvin Collaboration is intended to do is to look at both the data that we have already collected and also to look at other combination therapies of our FAK inhibitors with other drugs or immunotherapies in other cancers and so forth. So we're looking to expand the collaboration and look for other opportunities based on, both our clinical stage FAK assets and the Garvin's knowledge of FAK inhibition. So other combinations in other oncology indications, I guess, is the short answer to that question. What are your plans to fund the phase 2 clinical trials? As you can see, we have released our 4C today, and we're in constant discussions. The board and Amplia is in constant discussions with our corporate advisors. We are developing those plans right at the moment. Let me see what else. Does the use as a combination therapy accelerate the approval or commercialization process? I don't believe so. I don't think it would accelerate. If I think about a regulatory agency's view of these molecules, I think the FAK inhibitors are going to have to stand on their own 2 feet, whether it's a fibrotic or oncology indication that they're being developed in. However, the level of unmet need, I think, that exists for these therapeutics opens up some potentially interesting regulatory pathways for Amplia, particularly via the FDA, who have a number of expedited review pathways. And we're certainly looking at those for our products, and we'll be looking at whether any of those expedited review pathways apply to us and whether we can successfully receive them. Another question here is, what are your thoughts on the differing response, NASH symptoms and fibrosis for the 2 Amplia drugs? Interesting point. I'm not sure that the differences we're seeing in those experiments are actually significant when it comes to comparing AMP-945 to AMP-886. The two drugs themselves seem to have pretty similar activity in the NASH model. So I'm not sure whether or not there is actually a significant difference between the 2. Another question is orphan drug designation an option for any of these indications? Yes, it is. Thanks for the question. In fact, we have received orphan drug designation for AMP-945 in both pancreatic cancer and in idiopathic pulmonary fibrosis. So early last year, we went to the FDA and asked them to award us or sought approval for orphan drug designation, and we received that. I think I've addressed all the questions that we've received so far. I'll just give it a few more seconds in case anyone's got a question that they would like to ask that I haven't addressed yet. Okay. So nothing further is coming in on the questions. So I think at this point then I'd like to thank everyone for joining our webinar today. I sincerely hope that it's been informative. As we move forward with our development of Amplius FAK inhibitors, we'll be sure to keep you informed of any future developments. So at this point, I'd like to close the webinar. Thank you again for your attendance and wish you all a good day. Thank you.